Hello everyone, this is Nick Cap from your microbiology class. This is the third and final lecture for the module number 2, which we're looking at bacterial taxonomy, microbial diversity, and then basically eukaryotic microbiology. Again, like all of these lectures are meant to go kind of relatively quick and meant to supplement material that we're going over the book and to give you an idea of what you need to know in the book. Again, like all of those here, what we're going to try and do is give you a smattering of what some of these microorganisms are and kind of what they're capable of. And we will talk about that a little bit. Like I said before on all of these, that there is going to be a review document that you will have, and that will give you a list of the microorganisms that you absolutely must need to know. But at this point here, with the slide here, I'd recommend that you do record kind of what you see. Again, just like we march through the different types of microorganisms that or bacteria that are out there, we're going to be marching through the eukaryotes. And I want you to know the general classifications and some examples of each of those. Again, eukaryotic microorganisms are a fairly rare infection event in first world countries. We can typically avoid them through our behavior. We typically don't eat where we go poo. We tend to have sewage systems and stuff like that. But that doesn't trivialize what these diseases that these organisms can do. And again, diseases like malaria that I have here can cause over 500,000 deaths a year. And so, again, we may say kind of why we don't stop this is that tends to be people who don't make a lot of money. And so they can't pay us a lot of money. So we're able to get rid of those diseases in our first world countries, but they tend to be endemic in kind of third world countries where they don't make a lot of money. Groups like the Gates Foundation have been really good because they will pay people to do this research in order to try to get rid of it. But again, a company that needs to make money will tend to shy away from these, even though they kill lots of people, because it's basically just the people wouldn't be able to afford what they can do. Again, we're going to look at what a protist is. That's a historic term. It's basically, it used to be what we call I called it a junk drawer classification. And so anything that wasn't a plant, an animal, a bacteria, would go into the protist. They are eukaryotic, and for the most part, they are single-celled most of the time during their life cycles. And so the example that I gave you here, malaria, again, is a microorganism. It tends to live in the blood. So here is the malaria kind of living in the blood. And something relatively simple like having a mosquito net will actually prevent you from getting this. Another example, and again, this is looking at a diagnosis. This is an example of ringworm. It's not actually a worm. It's a fungal organism that actually kind of grows on the skin. Dermatophytes, we call them. Giardia lambda. I've had these guys here before. Very nasty diarrhea, etc. And so again, it's a single-celled organism. Although if you look at here, these guys are kind of quite complex in their structure. Second thing is that we may want to know is a little bit about the life cycle. And so what goes on in a life cycle, in a human being, a life cycle is whatever happens from when you are born to when you are able to kind of reproduce. And so the Eimeria consists of oocytes that are shed in the feces and may cause disease when ingested by basically a new host. And so what's really kind of interesting is that these organisms need to live in several different other organisms in order to complete their life cycle. And so what we can do here is these may be really difficult to treat in something like human beings, but if we can get rid of kind of one of these other stages in their life, like reducing mosquitoes, etc., we can greatly reduce the number of organisms that are there. Some of the basic cell body plans that we have are things like ciliates, like the paramecium, the amoebas, the amoeboid movement, and euglena, this particular flagellated structure. And so those are all examples, and the paramecium, amoeba, and euglena are typical organisms that you should know. And so in the domain Eukarya, we have a number of groups that are there, and again, we would have This is a proposed classification of the domain Eukarya. It's based on evolutionary relationships, divided into 6 supergroups. With each supergroup, there are multiple kingdoms. The dotted lines indicate that suggest it. So, again, if we look at it, so here's the algae and the plants. Here are animals, fungi, kinoflagellates, slime molds, etc. And these are all organisms. And I'm not going to test you on those too much. But some of the organisms that you definitely need to know would be the Giardia. You need to know what that is. And so here we give some of the supergroups, we give some of the subgroups that are in them. I'm going to name and I'm going to give you some of these organisms. And again, mainly we look at those because they are clinical samples. And so here's a chart on here. And so the amoebozoa use amoeboid movement, and Dictyostelium would be the slime molds, Fuguloseptica. Again, these are 2 examples that you would know. The plastids, Epicomplexans, which would, again, here you may recognize this, the same micrograph. This is the parasite that causes malaria, which is pretty good to know. Blantidium, again, a lot of these organisms basically cause fever, feeling weak, and kind of diarrhea, and they cause a lot of the same things. And so how these guys are recognized is by a clinician taking, let's say, a stool sample and looking at them underneath the stool sample and seeing if you can see anything. The paramecium has this kind of great structure, this great groove-like structure that they use to feed, and we'll probably see some paramecium on slides. And again, the water molds that we see, and here's a water mold that's taken over kind of an insect. The excavata have complex shapes and they have structures that look like they're kind of excavated inside of there. Trypanosoma bruceri, which causes the African sleeping sickness, and again spends some of its life cycle in a fly. So this goes from a human being to a fly, etc. And so I'd have a good idea of what this is. And these are all diseases that we'll look at a little bit later. We'll get a chance to look at it in class. Again, this is the pinworm we're looking at. So classes of nematodes. This is an odd-looking kind of nematode, but again, these are non-segmented worms. The flatworms, again, the beef tapeworm that we would have. It's interesting, the skullex, which is the head-like structure, and so these hooks attach onto the intestine and they feed on food as it flows by. Again, you need to know some of these life cycles that they do, and here's a life cycle of the tapeworm. And again, what we do with these guys here, how we've gotten rid of a lot of the tapeworms in human beings is, number 1, we cook our food, make sure that it's fully cooked. And number 2, we treat our farm animals to kind of get rid of some of those diseases. The guinea worm is an example of a worm. We'll take a look, and so this is kind of somebody's foot, and they're kind of pulling this worm out over time. The fungi are kind of interesting in that they are organisms, saprophytes, that basically live off of kind of dead things. But again, where they're really good in that they can grow really fast and that they have a capacity to metabolize almost kind of everything that's out there. And so again, you should know what the hyphae of these organisms look like and what yeast cells. The yeast are basically single-celled microorganisms. Histoplasma, which occurs in animal feces, and you can get this in barns and in various places. There's various fungi. Fungi are kind of interesting because they can actually degrade our crops. They can degrade cotton, some of our clothing. They can cause disease. There's a number of different things that we have. And so I do want you to know the Aspergillus, Canidia albicans, and know some of these and kind of what they do. They produce these ascospores. There should be 1, 2, 3, 4, 5, 6, 7, 8 inside there. These are the results of meiosis. So they were used to study the cell cycle kind of early on. Micronium and microchronis, and again, those are disease of the skin. The algae include things like dinoflagellates that can actually cause, along with the cyanobacteria, can cause kind of things like the red tide and stuff like that. These actually can fluoresce sometimes, and you'll see those light up at night. Again, I do want you to know, take examples of these organisms. Again, there are no parasitic forms of these, but you do need to know some of their life cycles. This is an example of Volvox. They're beautiful. If we're able to see some underneath the microscope, you'll be able to get pictures of them. The other thing that we want to talk about is basically symbionts, or basically two organisms live together. They can survive without one another, won't necessarily die, but they're not do it as well. In our campus, if we were up on campus, we would see lots of these. Is that where we have a fungus is able to produce a structure, whether it's able to grow the algae inside of it. So the fungi gets water, gets nutrients from its environment, and it shades the algae a little bit. It gives these nutrients to the algae, and then the algae release sugars that the fungi can use. And then we have some pictures of some of the fungi if you were able to get onto campus, although these aren't pictures from our campus. But that's basically about it. So we have now covered microbial taxonomy. We've looked at the diversity of prokaryotes and eukaryotes. We've looked at the diversity of archaebacteria. So it's now your possible time to take the quiz and participate in the discussion board, and we will have some activities in class where we will look at these and we'll report out these a little bit more. It's been my pleasure working with you. This is Nick Cap out for microbiology.